Okay, so let's start with the histology of liver. Now, what you have understood that day? What you have understood that day? In GIT, okay, 4 basic layers are there which we have studied. Okay, what are the 4 basic layers present in GIT? Mucosa, submucosa, then muscularis externa, serosa or adventitia. Okay, so those are the 4 basic layers which you have studied in GIT. That means we have seen the structure of esophagus also, then a stomach, after that small intestine and large intestine, where we have covered the appendix also. Okay, so during that you have seen all those layers were present, but there were some modifications, right? But here what is the difference? Here you can't write directly mucosa, submucosa, all those layers are not there. Okay, some changes are there. Okay, though these are you can't say liver is a part of GIT. This is an offshoot of GIT. Okay, the liver jo hota hai, this is offshoot of GIT. Then pancreas, this is also offshoot of GIT. After that gallbladder, this is also offshoot of GIT. Means they are accessory organ for GIT. Okay, so whenever certain organs are there, okay, at that time, how you have to study the microscopic structure, how you have to study, for that day I have explained, the concept should be clear. First of all, if you take any organ, okay, especially around the GIT, I am talking about GIT, any organ, for example liver, pancreas, okay, especially if you focus on this, what you will see that that particular structure is made up of 2 components. What are they? Stroma and parenchyma. Okay, it is made up of 2 components, stroma and parenchyma. So stroma में क्या रहता है, parenchyma में क्या रहता है? Now before going to the stroma and parenchyma, another important part, mostly the organ is covered by a connective tissue covering. Okay, that is called as capsule. Overall it is called as capsule, connective tissue capsule. Okay, for example, in case of, I hope you have studied kidney that day. You have seen the kidney, you have studied the layers of the kidney. Okay, तो पहले क्या रहता है, which is the innermost layer? Renal capsule. ठीक है, तो similarly in case of liver also you have seen what is the, which capsule is there? Glisson's capsule. Okay, so see, similarities are there. Next, I am not telling that kidney or liver ka microscopic structure same, it is not same, completely different, but I am giving an example. Next, so connective tissue covering will be there. After that, what will be done? From that connective tissue covering, it will send septa, okay, inside the substance of the gland. What it will do? It will divide it into small lobes. Okay, it will divide it into small lobes. What you have to do? You have to take one particular lobe and then you have to focus what exactly you can see inside the lobe. Okay, so the structure of one lobe is similar to the other lobes. Like this, we have to study the microscopic structure. This is the concept. Okay, now sometimes one lobe is there, further it is divisible into smaller lobules, which is present in case of liver. Okay, in other organs it is not there, only lobes are there. We will be taking one particular lobe and we can study the entire microscopic structure. Okay. Next point, as I have mentioned, that if you see the entire structure, entire organ, entire gland, it is made up of 2 parts. What are they? Stroma and parenchyma. Okay. What is the meaning of stroma? Supporting part. Okay. Stroma, what is the first letter? S. Supporting का first letter क्या है? S. So S for S, stroma means supporting. Okay. Next, what is the parenchyma? Secreting or the producing part. So here also parenchyma, P, producing part, first alphabet is P. So P for P, like this you can remember. Okay, so जो parenchyma होता है, what is the main function? उसमें particular tissues रहेगा, cells रहेगा, which will do the production, which will do the secretion. Okay, and that is specific for each and every organ. Right? So this is the concept, how to study those particular organs or structure. Okay, so as I said, you can't compare with the basic layers which we have discussed in case of GIT. Okay, it is totally different. So though you have understood now, see, this is a revision. I was expecting that you have studied all those things. Now in the histology of liver, so how we will study the histology of liver? First we will see that what is there in the stroma, what is there in the parenchyma. Before that we have to know that the entire liver is surrounded by a connective tissue capsule that is called as Glisson capsule, Glisson's capsule. Next, I hope you know that liver is a intraperitoneal organ. Okay, liver is a intraperitoneal organ. That means which peritoneum will be attached to the liver? Visceral peritoneum. Visceral peritoneum will be attached to the liver. Okay. Now, deep to that, okay. First, visceral peritoneum is attached to the liver. Deep to that, what is attached? The Glisson's capsule is attached. Okay. Which is covering the entire liver. So, the liver is an accessory gland for gastrointestinal system, which is made up of stroma and parenchyma, as I have mentioned. Now, the outer surface of the liver is lined by serous coat from visceral peritoneum. That means the peritoneal covering of the liver. Deep to that or beneath that serous coating, what we are getting? We are getting a connective tissue capsule that is called as Glisson's capsule. Okay, which is covering the entire organ. Along with that, it extends into the inferior as numerous branching septa and trabeculae. Okay, that means from that particular capsule, Glisson's capsule, the septa is going inside the gland and it is dividing the gland into or dividing the organ into different lobes. Okay, further that particular septum will branch again to further divide it into lobules. Okay. This is how the branching pattern of the septum is taking place, which is dividing the liver into smaller parts. Next, along with the septum, what is going in? Blood vessels, lymphatics, nerves. Okay, these are going in along with the septum because this also has to reach the various part of the liver. And liver is a highly vascular organ, you know that. Okay, so through that septum, branching pattern of the septum, blood vessels are also going in. Next, in the stroma, what exactly you have to know? So as I said, the stroma is the supporting part. So in the first supporting part, we are getting the Glisson's capsule. Okay. Next, the septa which is there, it is carrying blood vessels, nerves, lymphatics along with it. So those will come under stroma only. Okay. So as you can see, the septum from the Glisson's capsule branches extensively and divides the parenchyma into basic structural unit that is called as classical liver lobule. So first branch which will go, it will divide into lobes. Further it will branch, the septum further will branch to make it smaller lobules. These are called as classical liver lobule. Okay. Next, another part is there which is supporting the parenchyma, that is the reticular fibers, which are also called as type 3 collagen fibers. Okay, they are in the form of network like structure or also called as reticulum, okay, which will support the parenchyma. Okay, that is present inside the liver. Okay, so those will come under stroma or the supporting part. So supporting part mein kya rehta hai? What are the things present in the supporting part? First is the Glisson's capsule. Then the septum. Okay, septum means one, in case it will be septa, because from Glisson multiple septa are going inside the liver. Once it is going inside the liver, along with that what is going? Blood vessels, nerve, lymphatics are also going in. So they will also come under stroma of the liver. Okay. Next, what is present? Next is the reticular fibers. Okay, so these reticular fibers, how they are arranged? They are arranged in the form of network or also called as reticulum, and they are supporting the parenchyma. Okay, so this is how the supporting part of the liver is organized. Next is the parenchyma. So in parenchyma we are getting the hepatocytes, along with that other cells are also present. Okay, but mainly we are getting hepatocytes. Now hepatocytes are the cells of the liver. Now in this case how they are arranged? They are arranged in the form of laminae. Laminae का मतलब क्या है? Layers. They are arranged in the form of layers. Okay, उसमें एक ही layer का cell रहता है, not multiple layer. But the way they are arranged, it looks like they are in the form of layer. That's why it is called as hepatic laminae. ठीक है? Next, another important part of the parenchyma is that जो laminae होता है, suppose one laminae is there, another laminae is there, in between both the laminae or adjacent laminae, what is present? Sinusoids are there. Okay, then they are also called as hepatic sinusoids. In between the laminae, what is present? Hepatic sinusoids are present. Okay, so hepatic sinusoids, what are these hepatic sinusoids? No, sinusoids directly, they are not blood vessels. Okay. Yes. No, that is the exception in case of liver. You are correct. Okay, but this is the exception only in case of liver. So I don't know whether you have studied about the sinusoids or not. Sinusoid are nothing but the type of a capillary. Okay, sinusoids are nothing but the type of a capillary. Capillary का 3 type होता है. Can anyone tell me what are the 3 types of capillary? One is continuous, one is fenestrated, and another one is sinusoid. Okay, also called as discontinuous. Continuous, fenestrated, discontinuous are also called as sinusoids. Okay, why discontinuous? Because here the endothelium cells are not continuous with each other. Suppose एक endothelium, इसके बाद थोड़ा सा gap है, इसके बाद और एक endothelium. That's why it is also called as discontinuous. Okay, so these are the sinusoids, and in sinusoids the flow of the blood, the velocity of the blood is very less. Okay, so hepatic sinusoids, so where they are present? They are present between the hepatic laminae and also between the cords of the hepatocytes. Okay, so in between the hepatic laminae and the cords of the hepatocytes, that means the arrangement of the hepatocytes inside the liver. Now, these particular sinusoids are lined by endothelial cells and the Kupffer cells. Kupffer cells are specialized cells which are present in that particular area. They are having phagocytic functions. Okay. Now, adjacent sinusoids communicate with each other so that the intersinusoidal pressure can be maintained. Now, the blood from the sinusoids open into the central vein occupies the center of the lobule. Okay, this is the direction how the blood is flowing inside the sinusoid. Okay, that means from sinusoid blood will go into the vein which is present at the center of the lobule. Okay, that's why the direction is also called as centripetal direction, means towards the center. Now finally the central vein will drain into the hepatic vein, and hepatic vein will drain into the IVC, systemic vein. Okay, this is how the blood is flowing inside the liver. Now if you remember, I have shown you this diagram that day. Now in this diagram you can see this is the septum. ये देखो, this is the interlobular septum. Okay, interlobular septum. That means in between two adjacent lobule. Okay, the septum which is present, it is the interlobular septum. और ये कहाँ से आता है? From where the septum is coming? It is coming from the Glisson's capsule. Okay. Next, so you can consider this particular part as a lobule, okay, where you can see what are this? These are nothing but the hepatocytes. How they are arranged in the form of laminae, or they are also called as layer. In between the laminae, the empty spaces which are there, these are called as hepatic sinusoids. So this is one laminae, this is another laminae. In between this, the space which is present, this is called as hepatic sinusoids. Okay, so you can see multiple hepatic sinusoids are there. Sinusoids में क्या रहता है? Just now I have mentioned sinusoids are nothing but fenestrated endothelial cells. Okay, fenestrated होता है, along with that endothelial cells are present. Okay, and the endothelial cells which are present in the hepatic sinusoid, they are not continuous with each other. If you remember the blood vessel chapter which we have studied, especially arteries, vein, in the lumen I have mentioned that endothelial cells are present. Now those endothelial cells you have seen, they are continuous with each other. One endothelial, another endothelial, continuous. In between the endothelial cell, no gap is present. But here जो hepatic sinusoid है उसमें भी endothelial cells रहता है, but in between the endothelial cells gaps are present. Okay, that's why it is sinusoid or discontinuous capillary. Next, along with the in the hepatic sinusoid, along with the endothelial cell, another important type of cells are there, and these are called as Kupffer cells. Okay, these are called as Kupffer cells. Function क्या है? Phagocytic action. Next, about the hepatic lobules, okay, or also called as liver lobules. Okay, so in case of liver lobule, three types of liver lobules are there. That means if you see the substance of the liver, okay, already I have mentioned a connective tissue capsule is there, Glisson's capsule, from there septa is extending, multiple septa, okay, so particular lobe में divide होगा. Now suppose one septa again it is branching, okay, then what it will do, जो particular lobe था उसको फिर से divide करेगा in smaller parts and those are called as lobule. ठीक है. Now if you take one lobe, in that you should get three lobules, okay, three hepatic lobules. Now what are those three hepatic lobules? These are called as classical liver lobule, portal lobule and liver acinus. Okay, so these are the three lobules which are present. That means the substance, or you can say the parenchyma, is divided into three parts. Okay, what are these? These are the classical liver lobule, portal lobule and liver acinus. Okay, so the parenchyma of the liver. So parenchyma क्या था? In the beginning I have mentioned parenchyma means the secreting part or the producing part. Okay, which will produce or which will secrete something. Okay, we know that, I hope you have studied about the function of the liver. You know liver, what things are produced by the liver. Okay, so that particular part is divided into three lobule. So first, if you see the classical liver lobule, what is the shape of the classical liver lobule? It is hexagonal in shape. Okay, next in case of portal lobule it is triangular in shape, and in case of liver acinus it is elliptical, or you can do one thing, give a slash and write diamond shape. Okay, that will be better. Give a slash and write diamond shape, or you can write diamond shape also instead of writing the elliptical shape. Okay. Next, in classical liver lobule, at the center one central vein is there. Here at the central one portal triad is there. Now what is portal triad and what is present inside the portal triad, we will discuss. In case of liver acinus, the structure is a bit different. Here we are getting the two axes, one short axis and one long axis. Okay, and along with that we are getting three zones, zone 1, 2 and 3. Okay, which are related to the hepatic arteriole, which is supplying this particular area. Okay, so we'll see the classical liver lobule. Okay, what is present at the center, what is present at the periphery. Okay, now please keep that in mind. In case of classical liver lobule, first of all you have to concentrate on the hexagonal shape. Next you have to concentrate on the center, and then you have to concentrate on the periphery. Okay, so center में क्या रहेगा? Central vein रहेगा. Okay, central vein is present. See, one central vein is present at the center. From periphery towards the center, you can see the cords of cells or cords of hepatocytes are radiating from periphery towards the center. Okay, in between them, what you can expect, sinusoids are there. Okay, in that sinusoid, the blood which is there from the periphery, it is draining towards the middle, towards the center, where the central vein is present. Okay, next जो hexagon shape होता है, तो hexagon shape का at each and every angle, hexagon means angles will be there, 6 angles will be there. So on those 6 angles you will be getting portal triad. That means 3 things will be there which will be making the portal triad. Okay, and along with that you will be getting the connective tissue covering. Okay, so this is the basic structure in case of classical lobule. Okay, so in case of classical liver lobule, as I said, it is hexagonal in shape. What is present at the center? At the center we are getting the central vein. Okay, and it is surrounded by radiating anastomosing plates or cords of hepatocytes. Okay, now adjacent plates of hepatocytes are separated by sinusoids. Now between the adjacent classical lobule, a small quantity of connective tissue is present. Okay, within the portal triad. Now at the angle of the hexagon, portal triad runs into the portal canal. So at each and every angle, portal triad is formed, where the structures which are present, they has to travel. How they are traveling? They are traveling through the portal canal. Okay. Next, another important space is there which is related to the classical liver lobule, that is the space of Moll. Why it is important? Because from that particular space only the lymphatics of the liver starts. जो lymphatic drainage होता है liver का, the lymph जहाँ से निकल के आता है, so this is the part from where the lymphatic drainage of the liver starts. Okay, that is the space of Moll. This is also called as periportal space. The space of Moll is also called as periportal space. Now ये कहाँ पे रहता है? This is the space between the connective tissue stroma of portal canal and the outermost hepatocyte. Okay, so portal canal जो है, around the portal canal, the hepatocytes which are surrounding it, okay, and along with that the connective tissue which is present, in between that the space which is present, that is called the space of Moll. Okay, now it is the site for lymph formation in the liver. Okay, now if you compare it, the classical liver lobule which is hexagonal in shape, okay, in case of other animal, for example pig, it is clearly seen. Why it is clearly seen? Because the amount of connective tissue is more as compared to the human being. Okay, so it is difficult to identify the hexagonal shaped classical liver lobule in the histology slide, especially in case of human. But it is not like this, it is not present, it is present, but it is difficult to identify. Okay, so in pigs classical liver lobule are clearly seen because of thick connective tissue, but in human these lobules are not notably separated from each other. Why? Because the amount of connective tissue are less. Now here you can see the classical liver lobule. So what is the shape? Last bench. What is the shape? ये shape क्या है? कौन सा शेप? एक सेकेंड, कौन सा शेप है? हेक्सागोनल शेप. वेरी गुड. नाउ सेंटर में क्या रहता है? व्हाट इज प्रेजेंट एट द सेंटर? सेंट्रल वेन. ओके, नाउ फ्रॉम द सेंट्रल वेन, फ्रॉम द पेरिफेरी टुवर्ड्स द सेंट्रल वेन, व्हाट आर दिस? कॉर्ड्स ऑफ हेपाटोसाइट्स. इन बिटवीन द कॉर्ड्स ऑफ हेपाटोसाइट जो एम्प्टी स्पेसेस है, इसमें क्या रहता है? हेपाटिक साइनोसॉइड्स. हेपाटिक साइनोसॉइड्स आर प्रेजेंट. ठीक है. नाउ एट ईच एंड एवरी एंगल, व्हाट इज दिस? This is portal triad. At each and every angle you can see the structures which are present. These are called as portal triad. Okay. Now how the portal triad structures are passing? Through portal canal. Okay. Connective tissue canal रहता है, portal canal. Through that the triads are passing. The structures of the triad are passing. Okay. Now the blood which is coming through this particular blood vessel, okay, through hepatic sinusoids, first of all the arterial and venous blood will mix, right? After mixing, where they will go? They will go into the hepatic sinusoid. Through hepatic sinusoid gradually they are going towards the central vein. Okay, so this is called centripetal direction, towards the center. This is how the blood is flowing in the liver. Okay. Now in the portal vein, in the portal triad, you can see the reticle of the portal vein, hepatic ductule and hepatic arteriole. These three structure together forming the portal triad. Okay, so this particular structure, if you see for human liver, it is very difficult to identify. Means you can identify the portal triad, you can identify the central vein, you can also identify the radiating cords of cell. But adjacent portal lobule is very difficult to identify. Okay, why? Because of the amount of connective tissue which is very less. But in case of pigs, they are easily identified because of more amount of connective tissue. Okay. Now, along with the classical liver lobule, I have mentioned that an important space is there, that is called as space of Moll. उसमें उसका clinical important significance क्या है? What is the importance? Lymphatics of the liver start from the space of Moll. ठीक है. तो space of Moll कहाँ पे रहता है? Just see the space of Moll. Okay. So what is this? This is the portal triad. Okay. In portal triad you can see portal vein is there, hepatic arteriole is there, and bile ductule are there. These three structure is forming the portal triad. ठीक है. So around the portal triad, first of all you are getting a connective tissue covering. ठीक है. ये देखो, this is the connective tissue covering which you are getting around the portal triad. ठीक है. Next, outside of that, the portal triad is also covered by a layer of hepatocytes. You can see a layer of hepatocytes are there. ठीक है. Now in between these two, ये जो space है, this is called a space of Moll, from where the lymphatics of the liver begins. Okay. Next, in this particular diagram, you can see the direction, how the blood is flowing through the hepatic sinusoids and how it is going to the central vein. Okay. What about the bile? How it will flow? Opposite to each other. That means bile which will be produced by this particular cells will go like this. So the flow of the bile and flow of the blood is opposite to each other inside the liver. So we have seen that the parenchyma of the liver is divided into 3 lobules. What is the first one? Classical liver lobule. What is the shape? Hexagonal in shape. Next one क्या है? Portal lobule. What is the shape? Triangular in shape. Okay. So the portal lobule is triangular in shape. Now if you see the portal lobule, it is a triangular area of the parenchyma around each portal triad. Okay. Now here what you can expect, it is considered as the functional lobule as the bile duct lies at the center. Okay, that also in the portal triad. So portal triad का जो bile duct है, that will be present at the center. Okay, and at each and every angle you are getting one central vein. This is how the portal lobule is formed. Okay. So the central vein lies at the corner of each portal lobule and center में क्या रहता है? Bile duct रहता है of the portal triad. This is the structure of portal lobule. Now it is also called as the nutritional lobule of the liver, obtained by joining the central vein of 3 adjacent hepatic lobules. Okay. Next, portal canal. So portal canal already mentioned. Okay, through which the portal triad structures of portal triad are traveling or passing. Now these are the angular intervals at the periphery of each lobule filled by connective tissue. Now this particular canal is consisting of a radicle of portal vein, hepatic arteriole and interlobular bile duct. Interlobular bile duct. Okay. Next, if you see the blood supply, blood from the branch of the portal vein and from the branch of the hepatic artery enters the sinusoids, where the mixing of the blood is taking place, which is unique in case of liver. Okay. At the periphery of the lobule and passes towards the center, where it will drain into the central vein. From central vein, ultimately it will reach the hepatic vein, and from hepatic veins it will reach into the IVC. Okay. Now here you can see the portal lobule. So ये देखो, triangular in shape, portal lobule. Okay. Now at the center you are getting the. At the center, what you are getting? No. Portal triad is there. Portal triad is there, hepatic, but at the center what you are getting? Bile ductule. You are getting the bile ductule at the center, which is a part of the portal triad. Okay, and at each and every angle we are getting the central vein. See, we are getting the central vein. So this particular part which is present, this is called the portal lobule. Okay. Next, this is a 3D diagram of portal canal. Okay, this is a 3D diagram of portal canal. So see, this is the central vein and this is the portal canal through which the structures of the portal triad are passing. Okay, so this is, you can see this is a hexagonal shaped. So each and every angle you are getting one portal canal through which the portal triad, structures of portal triad are passing. Okay, now last one is the portal echinus. The portal echinus का shape क्या है? Diamond shaped or also called as elliptical shaped. So we'll keep it as a diamond shape. Okay, so it is a diamond shaped smaller unit of the liver which is supplied by one hepatic arteriole running along the line of the junction of 2 hepatic lobules. So one hepatic lobule, another hepatic lobule जहाँ पे join होता है, in between that one hepatic arteriole which is supplying both the adjacent areas of the hepatic lobule. Okay, that is the portal echinus. And if you see the shape of the portal echinus, it is diamond shaped. Okay. Now 2 central veins lies at the end of the portal echinus. Okay, at the end of the portal echinus 2 central veins are there. Okay, and this will constitute the long axis of the portal echinus. Okay. Now they are the functional unit of the liver and it is having 2 axes. The long axis of the portal echinus is formed by joining the 2 central vein and short axis by the adjacent portal triads. Okay, so 2 portal triad like this you will join, that will form the short axis, and long axis to central vein at the end. If you join, that will form the long axis. Okay. Next, the artery which is supplying that particular area, the portal echinus, depending on the area of supply, 3 zones are there. Okay, as you can see, liver echinus is having 3 zones. Okay, that is zone 1, zone 2, zone 3. And it is having some important clinical significance. Now what are that? First of all, if you see the zone 1, it is near the portal tract. Okay, so it receives more oxygen, but it is exposed to more toxin, which are coming into the liver with the help of portal vein. Next, zone 2, it lies between the zone 1 and zone 3. Okay, and in case of zone 3, it is near to the central vein and receives less amount of oxygenated blood. Injuries are more in this particular zone. Let me show you the diagram. Okay, so in this diagram you can see the portal acinus. Okay, so this is the hepatic arteriole which is supplying the blood, and this portal acinus is present in between the two lobules, two classical lobules. So this is one lobule, this is another lobule. In between the two lobules, this particular area is called the portal acinus. Diamond shaped hota hai. Now at both the end, what you are getting? You are getting the central vein. Okay. Next, if you concentrate over here, based on the hepatic arteriole which is supplying the portal acinus, 3 zones are there. As you can see, 1, 2 and 3. Okay, and all are having important clinical significance. Okay, next, the liver cells, as I have mentioned, they are placed, how they are arranged in the form of cords. Okay, the cords of hepatocytes are separated by hepatic sinusoids. Okay, now sinusoids are lined by discontinuous epithelium with discontinuous basal lamina. Next, stellate sinusoidal macrophages are also present. These are called as Kupffer cells. Okay. Now, along with this, another important space is there, that is called as space of Dis, or also called as perisinusoidal space. Okay, now what is this perisinusoidal space? So the sinusoidal epithelium is separated from the underlying hepatocytes by a small extracellular space, that is called a space of Dis. Okay, that means the endothelium which is present of the sinusoid and the hepatocytes, in between them a small extracellular space is there, that is called a space of Dis. तो इसमें क्या होता है, the hepatocytes which are present, okay, they are having microvilli, and those microvilli are projecting into the microvilli of the hepatocytes are projecting into the space of the Dis. Okay, this is the important significance of this particular space of Dis. Now, in case of liver, there is no significant blood-liver barrier. Why? Because the endothelium is not complete, the basement membrane or the basal lamina is not complete. That's why a complete barrier cannot be formed. Okay. Now, can you see the space of this? So, what are these? These are the cords of hepatocytes. Okay? And this is the endothelium. Now, in between the endothelium and the cords of hepatocytes, the extracellular space which is there, this is called as space of Dis. तो इसमें क्या होता है, the hepatocytes, the microvilli of the hepatocytes, they are projecting into the space of Dis. Okay, next, another type of cell which is present in the liver, okay, that is the hepatic stellate cells, are also called as Ito cell. They are mesenchymal in origin, okay, and they are present around the perisinusoidal space of Dis. तो ये जो particular perisinusoidal space है, or the space of Dis, in that another important type of cells are present, and these are called as Ito cells. इसका main function क्या है? They store vitamin A and lipids. Okay, next is the drainage system, that is the intrahepatic biliary tree. Okay, so extrahepatic one already we have discussed earlier. That means the bile which is produced from the liver. From where the bile is produced? No. Bile, from where the bile is produced? Tat. Who said gallbladder? Gallbladder से bile produce होता है. Liver से तो होता है. कहाँ से होता है liver का? Hepatocytes. From hepatocytes, the bile which is produced, what is the ultimate destination? कहाँ पे जाता है bile? Yes, कहाँ पे जाता है? Which part of the intestine? See, basic concept, I don't know why it's not clear. Jejunum में जाके कैसे jejunum में जाएगा directly? Which part of the duodenum? Second part of the duodenum. You have not studied duodenum. I know you have not studied duodenum. That's why. That day in the lecture hall also I have mentioned several times. while explaining about the extrahepatic biliary system, that time I have mentioned, and in the dissection hall you have seen same thing has been repeated for more than 3-4 times. After that also concept is not clear. तो और क्या करना है? What else we can do? कैसे जाएगा जेजुनम में? थोड़ा तो कॉमन सेंस अप्लाई करो. So the bile which is produced by the hepatocytes, ultimately they has to go into the second part of the duodenum. ठीक है? Because bile का function क्या होता है? हाँ, तो जेजुनम में जाके फैट कैसे ब्रेक करेगा? तो डिओडेनम में क्या होगा तो फिर? ओके, सो फॉर द इमल्सिफिकेशन ऑफ द फैट बाइल इज रिक्वायर्ड. ओके, तो व्हाट इज द end point means where the bile is going, that is into the second part of the duodenum. Now the flow of the bile, okay, is divided into two parts. When it is flowing inside the liver, then it is called as intrahepatic biliary system, or also called as intrahepatic biliary tree. ठीक है? Once it is coming out of the liver, then it is called as extrahepatic biliary system. As simple as that. Intrahepatic means inside the liver, extrahepatic means outside the liver. It has to come out from the liver. अगर liver से बाहर नहीं निकला, then how it will go to the second part of the duodenum? Okay. Now when the bile is coming out from the liver, okay, कैसे जाएगा? There must be some duct. It cannot mix to the blood directly. Blood के साथ directly mix नहीं हो सकता because it is not a hormone. Okay, तो कैसे जाएगा? For that ducts are required. So that duct which is present in the extrahepatic biliary system, that will conduct the bile into the second part of the duodenum. For that we have studied common hepatic ducts are there. Before that right and left hepatic duct, common hepatic duct, इसके बाद common bile duct. Okay, so that is the extrahepatic part. Now let's focus on the intrahepatic part. Intrahepatic part means, suppose the hepatocytes, whenever the hepatocytes are producing bile, okay, every day bile is produced in our body by liver. So once it is produced, it is poured directly into the bile canaliculi. ठीक है? It is poured directly into the bile canaliculi, and from the bile canaliculi gradually it has to come out. Okay, so I am talking about that particular pathway, and that is called as intrahepatic biliary drainage. Now intrahepatic biliary tree drains the exocrine liver, that is the bile, and conveys it into the extrahepatic biliary tree. How? With the help of right and left hepatic duct. Now the intrahepatic biliary tree consists of bile canaliculus. Okay, that is the canaliculi are merely the spaces present between the plasma membrane of the adjacent liver cell. So how the bile canaliculus are formed? These are formed by the spaces which is present between the plasma membrane of adjacent liver cells. Okay, where the bile is poured directly. After producing, bile is poured directly into the bile canaliculus. ठीक है? वहाँ से कैसे पास होगा? After that we are getting the canal of Herring, or also called as intralobular ductules. Okay, bile canaliculus के बाद what we are getting? Intralobular ductule. Now bile canaliculi drain bile into the intralobular ductule. Okay, now this particular intralobular ductule are lined by simple cuboidal epithelium. Next is interlobular duct. So intralobular duct will drain into the interlobular duct and then run through the interlobular connective tissue septa of the portal tract. Okay, this is from one duct to another duct, how the bile is flowing inside the liver. Okay, next these ducts are lined by simple cuboidal, and the larger ducts are lined by simple columnar epithelium. Okay, now let me show you the diagram. Now in this diagram you can see the intrahepatic biliary tree. So first is the bile canaliculus. Okay, so from the liver cell, that means from the hepatocytes, directly it will go into the bile canaliculus. After bile canaliculus you can see canal of Herring. Okay, then you can see interlobular ductules. Okay, then septal ductules are there, area ducts are there, then segmental ducts. Okay, from segmental ducts we are getting the right and left hepatic duct. As we are going downwards, you can see the diameter increases. That means the shape of the duct changes, the diameter increases. Okay, Once you are getting right and left hepatic duct, what it will do, it will come out from the liver. जो right and left hepatic duct है, it comes out from the liver, that also from the right and left lobe of the liver. Okay, after that you can see they are joined together to form the common hepatic duct. So from this particular part, this is the extrahepatic biliary system. Beyond that entire part, this is the intrahepatic biliary system, or also called as intrahepatic biliary tree. Okay, so in intrahepatic biliary tree you have to understand कहाँ से start होता है bile, मतलब कहाँ से secretion होता है. After that, through which pathway it is reaching up to the extrahepatic biliary system or into the common hepatic duct. Okay, and whenever it is passing through those duct, what is the lining epithelium of those duct. Okay, so if you see the beginning, that means into the canaliculi. In case of canaliculi, the cells are almost squamous epithelium. After that, the ductules, whatever we have mentioned, first in case of smaller duct, it will be simple cuboidal, and as you go towards the larger ductules, those will be simple columnar epithelium. Okay, so right and left hepatic ducts. So how the right and left hepatic ducts are formed? Interlobular ducts join to form the right and left hepatic duct. Later they will join to form the common hepatic duct. Now this common hepatic duct, it is outside the liver. So from there onwards we are getting the extrahepatic biliary system. Okay. Now in some cases duct of Luschka is there, also called as accessory bile duct. Now this is the flow, how the bile is flowing. So first you can see from where it starts from the hepatocytes. From hepatocytes, the bile which is produced, it will be poured directly into the bile canaliculi. Okay, so from bile canaliculi, it is going to the canal of Herring, or also called as intrahepatic ductules. Intrahepatic ductules, after that they are going to the interlobular, then they are going to the intralobular bile ductules, where it is lined by cuboidal epithelium, and larger one is lined by columnar epithelium. Then they are forming the right and left hepatic ducts. So this will join together to form the common hepatic duct. Like this, the bile is coming out of the liver. Okay. So two things which we have discussed today, if you have noticed, first the direction of the flow of the bile and the direction of the flow of blood inside the liver. Okay, next is the hepatocytes. So I have mentioned several times about the hepatocytes. Okay, so the liver parenchyma consists mainly of the hepatocytes. They are large polygonal cell. If you see the nuclei of the hepatocytes, spherical nuclei present at the center. Nuclei shows huge euchromatin because they are metabolically active. Next, sometimes hepatocytes are binucleated. Okay, lifespan is approximately 5 months. Regeneration capability, it is having high regeneration capability, okay, of the hepatocytes. Now cytoplasm is slightly acidophilic. This is the property. Next, the most important part of the hepatocytes are three surfaces are present of the hepatocytes. जो hepatocytes होता है उसका 3 surfaces होता है, and those are also very important. Now what are those 3 surfaces? First you can see sinusoidal surface, means which is facing towards the sinusoid, hepatic sinusoid. Then canalicular surface, which is facing towards the canaliculi, and intercellular, means towards the adjacent hepatocyte. So 3 surfaces are present, okay, in the hepatocytes. So what are those sinusoidal surface? It is facing towards the adjacent sinusoids. Okay. Now this surface of the hepatocyte shows microvilli, and that will project into the space of this, which I have mentioned earlier. ठीक है? तो ये जो microvilli रहता है, it will increase the surface area for transport of nutrients across the hepatocytes. That's why these microvilli are present. Next, canalicular surface. This wall forms the bile canaliculus. The diameter is approximately 5 micrometer, along with the adjacent hepatocytes, and these bile canaliculi are separated from remaining intercellular space by the tight junction. So in this particular surface, tight junctions are also present, and intercellular means towards the adjacent hepatocytes. So here you can see three surfaces. So see, this is the sinusoidal surface where microvilli are present. Next, this is the canalicular surface where it is forming the bile canaliculus, diameter 0.5 micrometer, where gap junctions are present. Okay, and this is the intercellular surface which is facing the adjacent hepatocyte. Okay. Next, these are the list of cells which are present. Okay, list of cells which are present in the various part of the liver. Okay, first the most important one. What is the most important one? Is the hepatocytes. Okay, so they are in the form of cords or plates. Okay, throughout the liver you can see hepatocytes are present. What is the major function? Production of bile, detoxification of toxic chemicals, storage of glycogen, etc. Next, cava cells. They are mainly present along with the sinusoids. Phagocytic action is the main function of the cava cells. Next, Ito cells are there, or also called as hepatic stellate cells. Star-like appearance are there. Where they are present? They are present in the space of disc. Okay, इसका function क्या है? Storage of vitamin A and lipids. Okay, now sometimes they become myofibroblast. Okay, in various liver disease, for example, liver fibrosis. Next, sinusoidal endothelium. That means the endothelial cells which are present. Okay, they are lining the sinusoids. Next, hemopoietic cells. They are only present during the fetal life. In adult, these cells are absent. Now this is the diagram, schematic diagram which you have to draw in your practical notebook. ठीक है? तो इसमें क्या दिखाई दे रहा है? What you can see? Classical liver lobules. Okay, ये देखो, this is one liver lobule, this is one liver lobule, this is one liver lobule. ठीक है? So this classical liver lobule are seen over here. Now in the liver lobule, what you can see? So you can, first of all, these empty spaces, जितना भी empty spaces दिखाई दे रहा है, this is nothing but the hepatic sinusoids. ठीक है? Next, you can see the cells which are present in the form of hepatic laminae. ये जो दिखाई दे रहा है, this is nothing but the hepatic laminae. Okay, next at the center ये जो white space है, this is the central vein. ठीक है? And you can see these are the portal triad. These are the structures of the portal triad. ये देखो, this is one portal triad, this is one portal triad, this is one portal triad. Okay, so this is under low magnification. That's why you can't see the individual structures of the portal triad. For that you have to zoom in and see under high magnification. So under high magnification you can see the structures of the portal triad. ठीक है? Now, can anyone tell me how you will identify or how you will distinguish between the structures of the portal triad? Any idea? Just see the hepatic artery, see the bile duct, and see the portal vein. Okay, so no.
Portal vein is elongated.
No. See, based on the, first of all, we can differentiate based on the lining epithelium which is present. ठीक है? Next, always it is not suitable to see the lining epithelium under low magnification, even under 40x also. So the best way is, depending on the size, you can differentiate. जो सबसे बड़ा वाला structure होगा, that is the portal vein. Okay, सबसे बड़ा वाला जो structure होगा, that will be the portal vein. Medium one will be the hepatic arteriole, and smallest one will be the bile ductule. Okay, so like this you can differentiate by looking at the size of the structures which are present inside the portal triad. So this is the schematic diagram for drawing. This is under low magnification and this is under high magnification. Okay, so now in the histolab, the slide which is available, in that it will appear like this. Okay, so I will request everyone to see the slide properly, because after that you have to identify the things in the under microscope. ठीक है? So क्या दिखाई दे रहा है? See, in the previous diagram, in previous diagram, can you see the classical liver lobule clearly? ये देखो, एकदम क्लियरली दिखाई दे रहा है. पोर्टल ट्राइड भी क्लियरली दिखाई दे रहा है. यहाँ पे देखो, it is not clear. You can't distinguish. So in the beginning I have mentioned, in case of human being, जो क्लासिकल लिबर लोब्यूल होता है, सपोज एक क्लासिकल लिबर लोब्यूल अनदर क्लासिकल लिबर लोब्यूल, दोनों के बीच में जो कनेक्टिव टिश्यू होता है दैट इज वेरी लेस, दैट्स व्हाई डिस्टिंग्विश नहीं होता है. हियर आल्सो यू कांट डिस्टिंग्विश, सब एक ही जैसा लग रहा है. इवन एट हाई मैग्निफिकेशन आल्सो इट्स वेरी डिफिकल्ट टू डिस्टिंग्विश. ओके, इट इज प्रेजेंट बट इट इज डिफिकल्ट टू डिस्टिंग्विश. तो यहाँ पे क्या देखना है, फर्स्ट यू विल सी द सेंट्रल वेन. कैन यू सी द सेंट्रल वेन? इसमें ये देखो, ये एक सेंट्रल वेन है, ये एक सेंट्रल वेन है. ठीक है. नेक्स्ट व्हाट यू हैव टू सी, यू हैव टू सी द हेपाटोसाइट्स. सो दिस आर द कॉर्ड्स ऑफ हेपाटोसाइट एंड इन बिटवीन दैट द एम्प्टी स्पेस जो है, व्हाट आर दिस आर द हेपाटिक साइनोसॉइड्स. ठीक है, hepatic sinusoids are there. Now if you zoom further, then only you will be able to distinguish between the structures of the portal tract. 10x में it is very difficult. Okay, under 10x it is very difficult to distinguish the structures of the portal tract. So under 40x it will appear like this. So see, these are the structures of the portal tract. You can see the hepatic artery, okay, you can see the portal vein and you can see the bile duct. Just now what I have mentioned, how you will identify? Depending on the size. Which one is the largest? ये देखो, ठीक है. मीडियम नेक्स्ट कौन सा है? हेपाटिक आर्टेरियल. ये देखो, व्हाट इज दिस? हेपाटिक आर्टेरियल. एंड विच इज द स्मॉलेस्ट? बाइल डक्ट ट्यूब. ओके, so we'll try to identify all those things under 10x, but as I said it is difficult. So for that we have to zoom under 40x, then only we can see all those things. Okay, now there are some important points. I will request everyone to note it down, these important points, because based on this you will be identifying the slide. Okay, so please keep that in mind. From now onwards all the structures whatever we will discuss, okay, those will be quite complicated. Okay, from now onwards whatever we will be discussing, जैसे you have seen the structure of liver, it's not that simple. Next when we will discuss the structure of kidney, it will be more complicated. Okay, so like this, as we will go further the structures will become more complicated. Now first of all, in this panoramic view, okay, first of all what you have to do, you have to see the hexagonal areas, that is called hepatic lobules. Now here also it is very difficult to find out the hexagonal area. So for that the best way under low magnification is we have to identify the central vein. ठीक है, तो मान लो एक central vein identify हुआ, then go towards the periphery. तो whenever you are going towards the periphery, you can see some structure. Focus in that particular part to identify the portal tract. ठीक है, like this, go in other direction, other angle to see the other portal tract. Like this, you have to complete one classical liver lobule. Next, irregular cords of cell seem to pass from this vein to the periphery of the lobule. Then these cords are made up of polygonal liver cells. These are called as hepatocytes. Along the periphery of the lobules, angular intervals are there filled by connective tissue where the portal tract are present. Okay. Now, what you can expect? Portal vein means tributary of portal vein will be there, hepatic arteriole will be there, and interlobular bile duct will be there. How to identify this? Just now I have mentioned how to identify this structure. Okay, I will request to write everyone to write that part also. How you will identify the structures present in the portal tract. ठीक है? So these are some important points, okay, which you have to know in order to identify the or understand the slide of the liver. Okay, when you have studied the specimen of the liver in dissection hall, have you seen the gallbladder? It was there with the specimen of the liver. I hope you have seen. Okay, very good. Now in case of gallbladder, it is made up of 3 layers. What are they? Mucosa is there. Now in case of gallbladder there are some similarities. Okay, जो gallbladder है उसमें similarities है. For example, you can see mucosa is there. Previously liver में कहीं पे mucosa था? लिवर में कहीं पे म्यूकोसा था? नहीं था. You have not seen any mucosa, submucosa. यहाँ पे थोड़ा सा similarity है. Okay, for example, you can see mucosa is there. ठीक है. Along with that, you can see lamina propria is also present. That means you can correlate with the structure of GIT which we have discussed earlier. Okay, so 3 layers are present in the gallbladder. What are they? First is the mucosa, then fibromuscular coat, then serosa or adventitia. ठीक है. सिरोजा एडवेंटिशिया का कॉन्सेप्ट सेम है, जहाँ पे पेरिटोनियम है सिरोजा, जहाँ पे नहीं है एडवेंटिशिया. ओके, so that I will not explain further. Now please concentrate on the mucosa. Do you remember the lining epithelium of GIT? Simple columnar epithelium. Okay, यहाँ पे क्या है? What is the lining epithelium? Okay, so as I said, some similarities are there. If you remember the lining epithelium of small intestine, ठीक है, just see similarities are there. Okay. Next, lamina propria. Now यहाँ पे क्या difference है? In lamina propria of the small intestine, we have seen that some amount of lymphoid follicles are there. Okay, for example, in case of ileum, pairs patches are present. ठीक है, but here जो lymphoid tissue है, it's totally absent in the lamina propria. Okay. Next, fibromuscular coat. So in the fibromuscular coat, what is present? Bundle of smooth muscle fibers. Along with that, collagen fibers and elastic fibers are present. ठीक है, and here the arrangement of the smooth muscle cells are irregular fashion. कोई भी definite pattern नहीं है. What about the GIT? How smooth muscle cells are present? Inner circular and outer longitudinal layer. That means a definite arrangement is present. यहाँ पे irregular arrangement है. That is the difference. Smooth muscle cells रहता है, but arrangement is irregular. And in between the smooth muscle cells, what are present? Collagen fibers and elastic fibers are present. So together they are forming the fibromuscular coat. Okay. Next, serosa. तो serosa already you know what is in the serosa. जहाँ पे peritoneum है, it will be serosa. Where the peritoneum is absent, it will be adventitia. Now, if you see the interior of the gallbladder, okay, mostly whenever it is empty, numerous muscular folds are present, okay, and due to the presence of the fold, the folds can be seen in the microscopic structure also, okay. So don't misjudge it as villi, okay. Appearance थोड़ा villi जैसा होता है, but villi नहीं है. Why appearance is like this? Because of the folds, muscular folds which are present in the interior of the gallbladder. Okay. Now lining epithelium, that is simple columnar epithelium, okay, with basally placed nucleus, faint eosinophilic cytoplasm. Epithelial shows numerous apical microvilli which will increase the surface area for absorption. Now gallbladder में absorption क्यों होता है? Why absorption is taking place inside the gallbladder? Any idea? I know villi is present, that's why absorption is taking place, but why? What is the location of the gallbladder? हाँ हाँ कहाँ पे, liver में कहाँ पे रहता है? Gallbladder fossa is there at the near the inferior border of the liver. Okay. Now what is the main function of this microvilli? So as I said, absorption is taking place. तो जो gallbladder है उसका function क्या है? Temporary storage of bile, not permanent. Whenever the bile is stored, okay, उसमें water content ज़्यादा होता है. So when it is stored inside the gallbladder, it concentrates the bile. How it will concentrate? It will absorb extra amount of water which is present in the bile. Okay, like this the concentration of the bile is taking place. That's why absorption is required. Okay. Next, the epithelium shows apical striated border because of the presence of microvilli. Okay. Next, lamina propria, so connective tissue and a number of fenestrated capillaries, that means blood vessels are present. Now, lamina propria of the gallbladder, as I said, doesn't have any lymphatic vessel. Okay, these are absent. Now, there is no muscularis mucosa and submucosa in the wall of the gallbladder. Okay, that's why in the beginning I have mentioned only 3 layers are there. Okay, now lamina propria may show presence of crypts because of the mucosal folding. Okay, crypts रहता है. Why the crypts are present? Because of the mucosal folding. That means you will see one fold is there and then again it is going down. So that particular part is called as crypt. Again it is one fold is there. Okay, so in between the 2 folds, the area which is present, this is called as crypt. And these are formed because of the muscular folds which are present inside the gallbladder. Next, fibromuscular coat is there. So fibromuscular coat As I said, bundles of smooth muscle cells are there, and in between that, collagen fibers and elastic fibers are present. Okay. Now, what about the smooth muscles? They are not organized like the gut, or they are not organized like the GIT. Okay. They run in different direction. That means irregular pattern. Now, there are few diverticular. Now, this is very important. There are a few deep diverticular there of the mucosa into the muscularis externa, or also called as fibromuscular coat. Now, these are also called as Rokitansky-Askov sinuses. Okay, so what is this Rokitansky-Askov sinuses? जो mucosa होता है, from the mucosa deep diverticular present, ठीक है, and that extend up to the fibromuscular coat or muscularis externa. Okay, they appear like gland. Okay, they appear like gland, but they are just extension of the mucosa. That is a unique feature in case of gallbladder. Okay. Next, lastly, the serosa or adventitia. Thick layer of dense connective tissue is there, where blood vessels, lymphatics, nerves, elastic fibers are present. Okay. Now, this is the schematic diagram of the gallbladder. Okay. Now, in this schematic diagram of the gallbladder, can you see the appearance? Due to the folds which are present, mucosal folds which are present inside the gallbladder. Okay. So, don't misjudge it as villi. Villi नहीं है. These are not villi. ठीक है. These are the folds. Now, in between the folds, ये जो areas है, these are called as crypts. Okay. These are called as crypts. Now, here you can see the lining epithelium. So, what is the lining epithelium? Simple columnar epithelium. That also having microvilli. Okay, where absorption is very important in this particular case. Next, ये देखो, this is the lamina propria. ये देखो, this is the lamina propria. Now, in the lamina propria, you can see blood vessels are present, but you can't see any aggregation of lymphoid tissue. It is absent. ठीक है. Next, this thick layer is made up of fibromuscular coat, where you can see smooth muscle fibers are there, and then you can see the elastic fibers. Okay, तो arrangement कैसे रहता है? First, smooth muscle fibers will be there, elastic fibers, then smooth muscle fibers, elastic fibers, like this stack wise. And here in case of gallbladder, जो smooth muscles है, उसका कोई particular arrangement नहीं रहता, irregularly they are arranged. Okay. Next, you can see Rokitansky-Askov sinuses. So, what are these? These are the diverticula from the mucosa. Okay, deep diverticula of the mucosa. And due to that diverticula, whenever the section is taken, it appears as gland, but actually ये gland नहीं है. And then ultimately here you can see the serosa. Okay. So, this schematic diagram, this you have to draw in your practical notebook. Now, lastly, this is the low magnification, and this is under high magnification. So, here you can see the mucosal folds which is present over here. This is the lining epithelium having microvilli. These are the crypts. These are the crypts. Okay. This is the lamina propria. This is the lamina propria. Now, wait. क्या हो रहा है? जाओ जाओ जाओ. बहाना खत्म नहीं होता. This particular part is the fibromuscular coat. Okay, now you can see the smooth muscle fibers. See, this is the smooth muscle fibers. Okay, this is the smooth muscle fibers. Along with that, you can also see the elastic fibers. Okay, and here this is the serosa. Now, in case of gallbladder, please keep that in mind. जो serosal layer है, that is quite thick. जो serosa होता है, in case of gallbladder, it is quite thick. Okay. So that is all about the gallbladder. Now we'll go to the lab and we'll see the slides.